As Barbara said, thank you, Barbara, for inviting me, and the steering committee, which I'm a member of, for inviting me. And so my job today, as Barbara said, I'm a neuroscientist, but I'm really a molecular and trained in molecular biology and biochemistry. So my job this morning is really to give you an introduction to biology, the molecular basis of learning and memory. And so this has been an interest of mine since high school. I was telling stories about my high school research project last night. I was in learning. And so what I've wanted to do my whole career is try to understand what changes in your brain when you learn something. How do you encode that memory? And then how does that memory last for decades? I mean, that's an incredible biological process. And so I'm going to go through research in our laboratories, but summarize the field as well. And if we... So many of you know the brain has billions of neurons. The human brain is estimated at about 100 billion neurons. And these neurons, just schematized here, each of these little circles is a neuron, is interconnected with other neurons to form very complex neuronal circuits in the brain. And these neuronal circuits really underlie everything we do, from being able to learn and remember, to have emotions, to have actions, to make decisions. So all our functioning of the brain and our mind is encoded in these circuits. Now, how do these circuits form? The next slide just illustrates two neurons. And these circuits form through contacts called synapses, which many of you are probably familiar with. And as I said, there are 100 billion neurons, but each neuron can have about 10,000 synapses. So each of these neurons are connected to 10,000 other neurons, and so you have quadrillions of these synaptic connections in your brain. So right now, these quadrillions of synapses are firing away in your brain. Hopefully I'm going to modify them a little bit so you can remember me or remember the talk, at least not in detail, but in hopefully some memories. And so these neurons communicate at these points of contact, and they communicate through, neurons work through electrical currents. So this neuron will receive 10,000 inputs. This will produce a action potential electrical current, which will run down the axon here. And then it's got to convert this electrical signal into a chemical signal, because there's a gap between these two neurons, and then convert it back into an electrical signal. And this is illustrated here. So this is a blow-up of this synaptic connection. And so an electrical signal action potential comes down here. It promotes the release of neurotransmitters, which are packaged in little vesicles, as shown here. And then these diffuse across the synaptic cleft, this gap here, and then bind to receptors on the postsynaptic cell. And then these receptors convert this chemical signal back into an electrical signal. So this is the process of synaptic transmission that's going on quadrillions of times in your brain right now. Now, this is not a static process. This occurs rapidly within milliseconds, and it happens very reliably, but it's got to be dynamic. It's got to respond to the environment. It's got to change as you grow, develop, as you age as well. And so this has to be what we call plastic. It has to be able to change and take inputs from the environment and modify it. And this is how we think memories are encoded. That memories change the synaptic connectivity in the brain to form neuronal circuits, a novel new neuronal circuit, which will encode the memory. And I'll define this a little bit more in the next couple slides. So neuroscientists for years had been looking for synaptic transmission that was dynamic, that was plastic. And one of the classic experiments from the 1970s was the first observation of this sort of plasticity in a brain slice preparation. So this is a slice of brain called the hippocampus, just schematized here. And the classic experiment is that people were measuring the strength of synaptic transmission in this area. So you stimulate an electrode here, and then record the synaptic strength in neurons far away. And this preparation of the hippocampus is very relevant because many of you know the famous patient called HM. HM had epilepsy, and he had surgery to remove the portion of his brain that was causing the seizures. And this turned out to be the hippocampus, and this was done in the 1950s. And it turned out that right after his surgery, he could never learn anything new. He could remember his past, but every day his doctors came in and reintroduced himself to the doctors because he didn't remember them from the day before. And this went on for 40 years. So HM actually recently died, but it really focused our attention to the hippocampus as a key part of the brain involved in learning. So in this experiment by Bliss and Loma, what they did is they stimulated and record, and here we're just going to, this is with time, this is synaptic strength, and you can see if they just stimulate and record, schematically you get a stable baseline, a stable synaptic strength. However, they found if they gave a very high frequency stimulation and then just came back a minute later, they saw that the strength of the synapses doubled within that short period stimulation. And this plasticity, this increase in synaptic strength, lasted for a couple hours. And in fact, you can do this in an attacked animal, and this synaptic strength will last for a year. Okay, so this is an incredible phenomena. Brief stimulation increases the synaptic strength, and it's very, very long lasting, lasting for up to a year, and could be involved, in theory, in coding and memory. In the same area, Mark Bayer's laboratory found the opposite form of plasticity. This is called long-term potentiation, or LTP. They found that in the same area, if you give a stable baseline, you get nice stable strength. But if you get a very low frequency stimulation, in contrast to the high frequency stimulation, you get a depression. So you get a decrease in synaptic strength that again can be very long lasting. So these two type of mechanisms can sculpt circuits in the brain. It can increase the connectivity between certain neurons, decrease connectivity between other neurons, and in this way, carve out this circuit that encodes a memory, for example. All right, so how does this occur? And this is what my career began, is to really try to understand this at a molecular level. So if you're looking at a synapse, you want to make this stronger, you could release more neurotransmitters. So you could, after LTP and increased synaptic strength, you could be releasing more neurotransmitters, giving a bigger response. Or you could, for example, the receptors, you could modify the receptors and make them either more abundant or more sensitive. And beginning of my career, I decided to focus on these receptors and try to understand how they're regulated and whether regulation of these receptors could be involved in synaptic strength, LTP, and learning and memory. So over the years, we've characterized these receptors in great detail. They're ion channels, and we've studied how they're chemically modified, so how they are modified by what we call post-translational modifications. So enzymes add little chemical groups to them, and these either so-called phosphorylation sites, palmitoylation sites, or ubiquitination sites can affect how these receptors work. They can make the receptors more sensitive, less sensitive, depending on the site and modification, specific modification. These modifications are very dynamic, and they have enzymes that remove them. So you can have this very dynamic on and off regulating the function of the receptor, and thereby making the synapse stronger or weaker. So that's one way to regulate receptor function. Now, another way is to regulate the density of receptors at synapses. And here's a schematic. As some of you may know, there are two major types of synapses in the brain, inhibitory and excitatory. So these increase the firing of this neuron, where this synapse will decrease the firing of this neuron. And they have different receptors. So these receptors are called glutamate receptors. These are called GABA receptors for their neurotransmitter. And these receptors are concentrated at the point of contact, at the synapse. So they don't distribute all over the cell. They know where to go. And this is really, again, a very interesting biological phenomena, because these receptors are synthesized in the same process in the cell body and transported out, but they have to know where to get targeted. They have to know which synapse to go to. And we now know this is not a static process at all either. So you can add receptors quickly, and that could strengthen synapses, or you can remove receptors, and that will decrease the strength of the synapse. So again, removing the receptors, you're going to decrease the synaptic strength involved. So my lab has been studying these trafficking processes, and it's very complex, extremely complex, and modification of the receptors, along with many, many labs across the world. So today what I'm going to talk about is the excitatory synapse, and really I'm not going to go into detail, but the subclass of receptors called AMPA receptors. These are the receptors that mediate 70% of synaptic transmission of the brain. So they're really the key molecule, key receptors involved in, we think, in modification of synaptic function and synaptic strength. Now, over the last 15 or 20 years, my lab and many labs across the world have come up with a model for how this classic long-term potentiation, or LTP, occurs. And that is due to the dynamic regulation of receptor targeting to the synapses. So when you induce LTP, which is thought to be underlying learning and memory, you actually recruit receptors to the synapse. They get inserted into the plasma membrane and then recruited to the synapse, increasing the synaptic strength because there's more receptors at this postsynaptic site. Just the opposite occurs, we think, in long-term depression, that during induction of long-term depression, you actually remove receptors from the synapse and decrease synaptic strength. So this is very complex. How does this happen? How does the cell know to add the receptors and remove the receptors? And what are the signaling mechanisms? What are the molecules that help sort of escort the proteins to the synapse or take them away from the synapse? And so we've characterized this in great detail. As I mentioned, receptors sort of look like this. They bind a neurotransmitter here. They have an ion channel down the middle. And what we characterize a lot of these chemical modifications shown here, and we characterize what they do to the receptor. But we also characterized a variety of proteins that bind to the receptor, and these proteins help escort the receptor to the synapse. So for example, when the receptor gets to the synapse, it binds to proteins that are in the synapse, and that stabilizes the receptors there and maintains a high concentration of the synapses, receptors at the synapses. So just an example of a couple of the modifications. We've characterized a lot of these post-translational modifications I'm not going to go into, but there's many different phosphorylation sites, palmitoylation sites. Each of these does something different. It affects the channel properties of the receptors or affects how they interact with these interacting proteins. But we've also characterized the receptors that, as I said, bind to the receptors, the proteins that bind to the receptors. And this is actually a simplified version of this complex. And so these proteins, as I said, are involved in sort of escorting the receptors as they come out of the cell body. They're transported out to the synapse. They're involved in coupling the receptors to the machinery that brings them back inside the cell, and involved in LTP and LTD. So today I'm going to talk about two just examples, two proteins we've characterized. One's called KIBRA and one's called SynGAP. And these are proteins that we identified in the 90s. And KIBRA, it turns out, is, as I'll show you, is really key for normal human memory performance. And SynGAP, as I'll tell you, is one of the very most common genes that is mutated in intellectual disability, autism, and as well as schizophrenia. So KIBRA we had cloned in the 1990s, and we had cloned a lot of these proteins. We had identified a lot of these proteins. And so it was sort of sitting on the shelf. Nobody was working on it in my laboratory. This is 1999. And then this paper came out in 2006, and it says common KIBRA alleles are associated with human memory performance. And this has been highly reproduced, and also KIBRA is protective against late onset Alzheimer's disease. Now, it turns out that these two different KIBRA variants, genetic variants in the human population, is very common. About 70% of you have what they call the smart allele, and about 30% of you have sort of the less smart allele. But don't worry about it. It's only, you can remember 7 words versus 10 words and a word recall. But we thought this was intriguing, and we had identified it part of this complex. This is what the protein looks at sort of a structural level, so you don't have to worry about it. So we started studying KIBRA and characterizing what it does. And I'm not going to go through it, but it turns out that KIBRA is involved in this complex, very complex trafficking of the receptor to and from the synapse. Now, one way we study this, we want to go from molecules to behavior. So the way we do that using animals is using genetic manipulation of mice. And so in this case, what we can do is use genetics to actually delete the KIBRA gene, so we can mutate the gene so it's not present, so the mouse doesn't have this gene at all. And then we generate the mouse. And it turned out in this case, in some cases when you do this, the mice will die or have developmental defects. But in this case, KIBRA knockout had a relatively normal behavior and normal development of the brain. So we could then test both physiologically and behaviorally whether knocking out this gene had any effects. And so we did the classic LTP and LTD experiments, and shown here, this is the wild type. You get this nice synaptic increase, potentiation. And in the knockout, you have a deficit in this potentiation. It's not completely gone, but it's definitely significantly decreased. And we saw the same thing in LTD. Here's the control wild type mouse, and this is the mouse with lacking the KIBRA gene. The LTD is decreased. Now, what about behavior? So we do a lot of behavior in the laboratories, a variety of different types of behavior. One is a fear conditioning, trace fear conditioning. And in this case, you take a mouse, you put them in a little cage and get them used to it. And then the next day, you present a tone, and then you give them a little shock. Now, before you say I'm an animal torturer, we do this to humans as well. So the shock is relatively minor, but they learn this very, very quickly. And what their response to the shock and the learning is they'll freeze. They'll just sit still. And then you can take them out and you put them back in this context, and then without even giving the tone or the shock, they'll freeze it because they'll remember it from the previous day. Or you could put it into a new environment and then present the tone, and then they'll respond to the tone with freezing as well. So I'm going to show you. This is just an example of the mouse in the cage. I'm not going to show you him getting shocked, but you take him out of this. So he's in there, and you train him. You give him the tone and the shock. And then you transfer him to the texting context. He's moving around. Then you give him a little tone. So the tone, no shock, and you see he freezes. And then the tone goes off. And he starts moving around. So we can quantitate this as shown here. This is the learning process. So here's the wild-type mouse. You see it rapidly learns. This is how much it freezes in response to the training. And here's the KIBRA mouse. So the KIBRA mouse learns, but it learns slower, but it does learn to the same extent on that first day. But the contextual fear, when you put him back in that shock chamber, the wild type will freeze immediately, where the KIBRA the next day forgets about the shock and does not freeze, as shown here. And when you present the tone. When you present the tone, the wild type mouse will freeze, and the KIBRA mutant mice does not freeze. So this is one example where we're trying to couple these genes that we've discovered through biochemistry and molecular biology to behavior. All right, so the second example is the SynGAP gene. And again, we cloned this in the 1990s. The green here are synapses, and the green is actually a fluorescent form of SynGAP. So you can see SynGAP is a gene that's highly localized at these synaptic connections in these neurons. Here's the structure of the gene, and I'm not going to go into the details of this again, but we know what SynGAP does. It's what we call a RAS gap. It regulates RAS signaling in the synapse, and this is a key signaling component that's involved in recruiting receptors to the synapse during LTP. And again, we can delete it. So if we delete SynGAP, actually, if you delete it, it's embryonic, or it's lethal. The mice die shortly after birth. But the heterozygote, if you only have 50% of SynGAP, it has profound effects on behavior and plasticity. So here is the LTP, the classic experiment. This is the wild-type mouse. You see nice potentiation. This is a heterozygote. So this is a mouse where only one allele of the SynGAP gene is deleted. So it's 50% of SynGAP, and you can see there's basically no plasticity at all. So this mouse is severely compromised in plasticity, and it turns out he's severely compromised in learning. And so here, another test we do is a working memory test, the Y maze, and many of you are familiar with this. So if you put a mouse in this chamber, it's got three pathways. And what mice are very curious, so they'll go down and investigate each component of the Y maze. And what they do is when they get to this point, they'll always go to the one they didn't go to before because they're very curious. And to remember that they have already been down this one and to go this way, they have to have an active working memory. So they have to know where they're going and where they've been in the past few minutes. And so you can score this, and it turns out here's a wild-type mouse. You can see that 90% of the time that mouse is always alternating between the three Y mazes, as shown here. But the mutant, the one that's lacking 50% of SynGAP, is severely compromised. So it's basically at chance level. Now this mouse has other defects in water maze, other learning paradigms as well. Now again, we characterized that it's in the 1990s or so, but recently human SynGAP mutations were found in intellectual disability. This is the first one in 2009. And it turns out there's now dozens of children who have been found that have this same mutation, various mutations in the SynGAP gene. And in fact, there's a association, Bridge the Gap SynGAP, and this is supporting these dozens of children who have mutations in SynGAP. And so we've been studying these mutations, how they affect SynGAP function, how they affect learning and plasticity as well. Now, even more interestingly, SynGAP now, in the last year or two, has been found to be highly mutated in schizophrenia and autism. And this is interesting because they're different mutations than the ones that are found in intellectual disability. So we're trying to understand why these mutations produce such a different phenotype, such a different change in cognitive function. Okay, so I think I have a few more minutes here. So finally, I want to tell you something we're doing which is out of the crazy and sort of the most fun thing we're doing in the laboratory right now. And that is actually to watch learning occur at a molecular level inside an intact, alive, behaving animal, mouse. So we can image receptors at synapses by, in this case, the red synapses are excitatory, the whatever color that is, I'm colorblind, the magenta or whatever, white sort of colors are the inhibitory synapses. So you can actually watch how many receptors are at the synapse by imaging. And this is done in a tissue culture dish. So we're just imaging this in neurons in a dish. But recently, there's a revolution in the way we can visualize brain, and we can actually visualize these receptors in an intact animal under a microscope. So we take an animal, awake, behaving animal, and you can sort of put them in this little tube, and then we have ways to engineer and make the receptors fluoresce, to be green. So we can make the receptors be green and the neurons, the structure of the neuron, to be red. And then what we can do is put the mouse under the microscope and actually train the mouse under the microscope and watch the receptors move around. So this, if we're really right, we should be able to see that receptors are getting recruited to the synapses during learning, and there should be tight correlations between this receptor distribution and learning. So this is an example image. So what we're doing is we're looking at the very top of the brain, right at the cortex. We're going to go with the microscope, we're going to focus down deep into the brain, about a couple hundred microns, and we're going to see the receptors and the neurons. So this is the top where all the synapses are. So the green is the receptors, the synapses, and the magenta is actually just shows the structure of the cell. So these are the cell bodies down about 200 microns into the brain. And this is a 3D reconstruction of that. And so again, this is a live animal sitting under a microscope. So we're looking at, here's the receptors up here, in the green, all the synapses are up here. Here's the cell body. And we can zoom in using the software and look in detail at the synapses that are located up here. And this is pretty crazy. I mean, if you told me 10 years ago we'd be able to do this, I would say, you're crazy. But so we zoom in, and here, this is a dendrite, and these are the synapses shown here. And the green is the receptors. So we can actually see receptors in great detail in this brain. Now you can do this over time too. So you can put the animal back in the cage, and then every couple of days bring it back out and image. And remember, there are billions of neurons. We can find exactly the same neuron and look at the same synapse we looked at 2 days ago. So here we can study the level of receptors at the synapse over a month. Now, what about when we induce plasticity or we do learning? And if we do nothing and just image over a couple hours, you see receptors that are relatively stable. This is the green or the white here. In contrast, if we induce plasticity, we start to see the receptors change. So here you see receptors increase in the spine, in the synapse, after induction of plasticity. So this is what we're doing. We're actually watching these receptors go into synapse, make the synapses stronger in real time as they're learning. So we can do this while, trial by trial as the mouse is learning. So what I've tried to give you a sort of a very broad idea of the biology of learning and what we're trying to do and try to understand learning. My motivation has always been basic science, that I really want to understand how the brain works. But this has obvious, translational possibilities. The most obvious are disease for intellectual disability, and we're actually trying, because we know the pathway, the SynGAP pathway, we're using drugs, actually statins that people take every day, affect this pathway. And so we're using that to try to cure the defects in the SynGAP mutant mouse. And this could potentially then be used in humans with intellectual disability, but also with autism and schizophrenia as well. Now, it's a little bit science fiction, but cognitive enhancers, although there aren't really any good ones now, there will be. And so I think it's going to be an issue for the future, ethical issue for the future. When do we use cognitive enhancers? When do we not? Certainly there are better. There's going to be a lot better drugs for kids with ADD and other learning disorders. Whether we use them or not, I think it's going to be up to us. But the translation of trying to understand, knowing the basic science and the pathways involved in learning, really give us the opportunity to translate these findings into helping people learn. And also, the most obvious, of course, is Alzheimer's disease and age-related dementia. So just finally, I didn't do any of this work. I sit in front of a computer all day. And so this work, the work on KIBRA was done by Laura McCooch, a graduate student, ex-graduate student, and Lenore Volk. Lauren is now at Ursinus College, a teaching assistant professor. Lauren has her own laboratory at UT Southwestern. And the work on SynGAP is done by Yoichi Araki, a very talented research associate in the lab. And this is the rest of the laboratory doing related work. The funding, I've had long-term funding from Howard Hughes Medical Institute, National Institute of Neurological Disorders and Stroke, National Institute of Mental Health, the Simons Foundation, who fund autism research, and the Stanley Foundation, which funds schizophrenia research. So thank you very much. I'm happy to take questions.
Yeah, we've got about close to, I mean, we're running a little bit late, but that's okay. I'd say maybe eight minutes for questions.
Good morning. It's known that traumatic events when one is a young child can have major lifelong effects. Could you comment on this from your perspective?
Sure. So, so we've been studying trauma-induced memories as well, fear conditioning is a form of trauma. So if you present animals with this shock and the tone, they'll remember that for a month, and you can sort of reverse that type of learning through extinction process, which is sort of like behavioral therapy, basically, that you exposure therapy. What you do is you take the mouse and you put them in the environment, and you maybe present the tone repetitively, like 20 times in a row without a shock, okay? And so then they learn to ignore the tone, so they won't freeze anymore. So we studied this and we try to understand what that process is, the extinction process is, and it turns out it's LTD. So when they learn the trauma, the fear association, they potentiate the synapses, they make them stronger. And during the extinction process, they actually make them weaker by reversing that through an LTD-like mechanism. And we found it's a little complicated, but we found a way to avoid the relapse that is usually associated with exposure therapy. So exposure therapy will help humans and mice for a couple weeks or a month or so, but there's relapses are common when they're hear loud noise or something like that. But we found a pathway that we think truly erases not the memory itself, but the fear association with the memory. And so this has got to do with a type of receptor called the mGluR1 receptor. And so we're developing drugs against mGluR1 for treatment, potential treatment of post-traumatic stress. Now with kids, and the idea there is actually, well, I won't get into the weeds, but obviously kids, who are exposed, or vets who have these traumatic events, this therapy could potentially be helpful. So again, here's a basic research we've done. Our observations have been also seen in humans and in an experimental situation. So understanding the basic biology has led us to a translational drug, hopefully. Yeah, so we have not done that, but yeah, repeat the question. Are there epigenetic factors that regulate the expression of these two genes or the process in general? So, I mean, this is really critical because environment's going to affect, has a big impact besides the genetics that I've talked about. And So if you, and as I said, the level of SynGAP really affects, how the mouse learns. If you cut it down 50%, it doesn't learn very well. So in theory, epigenetic regulation of the expression level of SynGAP could be critical, but we haven't studied it.
Thank you very much for such an intriguing presentation. My question is whether what you showed us about it applies only to the hippocampus. Is it particular? Are there differences in other areas of the brain? Does learning take place in different ways?
Yeah. So a lot of the data I showed you was hippocampus, but this type of plasticity occurs basically everywhere in the brain. And this is the major form that we study. There are other forms of plasticity that also occur. And in fact, that the images I showed you in the mouse, that was actually in the cortex. So these types of plasticity occur everywhere. And that, of course, brings a worry about specificity. If we develop drugs, we want to not inhibit plasticity everywhere. But we're trying to identify brain-specific or neuron-specific mechanisms of plasticity so we can go in and target those for modification or for therapeutic purposes.
Thank you. I was wondering about resolution of these effects in time and whether or not say, learning in the classroom, there's a lot of talk about repetition and rehearsal, and revisiting the same material again. So are we seeing that kind of behavior or activity is necessary because of the way the brain learns or the time scale on which the brain learns? Is that why we need to do that?
Yeah, so we can study that. So all that's reproduced in animals. So if you do repetitive training over several weeks, it's much better than doing mass training in a couple days. But when you start looking at the mechanisms, you can also see that it occurs at the molecular level too as well. So you're seeing that there's this process of reconsolidation. So when you learn something, it will fade with time. But if you retrain, you get a reconsolidation, a strengthening, and you can measure that as a strengthening of the synapse. And that's one reason we want to image, because we can train a mouse. We can image the changes that occur. Wait a week or 2 weeks, see what happens to the receptors, retrain the mouse, and see if what happens to the receptors. So really to dissect the mechanisms involved in these, consolidation and different types of learning, different types of basically teaching the mouse.